taxonomy, biology of property, and political evolution.
And good afternoon, everyone. Can you hear my voice? Oh, hi. Okay, my name is Yugo, as he introduced, and I'm a Japanese researcher and working at the Season Partner of the Botanical Garden, China. And today's talk is one of my postdoctoral research tema. So and before I start talking about what the genus Najas or what I'm interested, I will give you a brief prologue and then move on to the specific topic. And this one is the famous map for those who are interested in conservation of biodiversity. And this one shows the biodiversity hotspot in the world, including 34 spots, including, for instance, our country, Japan, and New Zealand, for instance, and here we are indicating the biodiversity hotspot. And from the same website, it is said that there are places on Earth that are both biologically rich and deeply threatened. For our own sake, we must work to protect them. Oh, thank you. And we have a question on which perspective do we work? As long as I'm a taxonomist, I think we have something to do from the point of view of taxonomy. So here, we can see some important paper entitled The Law of Taxonomy in Conservation Biology and The Law of Taxonomy in Species Conservation. The other one is Conservation of Biodiversity through Taxonomy. And I picked up some phrase, for instance, I'm not sure from which one, but taxonomy is the foundation of biodiversity science. And another one, new knowledge of existing species and discovery of undescribed species are urgently required. So, we would say taxonomy links biodiversity, and based on the which conservation takes account. So we have something to do. There's something we can do. Now I will show you my species of interest. I have been working on the taxonomy systematics of aquatic species, water plant, and which is in some lineages in the angiosperm phylogeny, such as the basal one, Lepiaceae, and then some of the monotype species, Aristotales, and then Fabaceae, some of the Fabaceae species of aquatic. And today's talk is about genus Najas, which is among the species of Aristotales monotypes. Najas is a species of aquatic monotypes. So what the species look like? And here I picked up important diagnostic characters of genus Najas, such as leaf texture, shade code, leaf sheath, female flower, leaf surface, and the male flower space. It's not easy to make it. For instance, using leaf texture, you can divide the species into subgenera Najas and Carolina over there. And another important character is the seed code. I don't know if you can see this, but for instance here, the broadly rectangular morphology, which is ladder-like arrangement, is different from this kind of randomly arranged hexagonal seed code morphology. And there's other thing, like leaf sheath is sometimes used for identification, but sometimes intermediate, so difficult to use. And here, female flower number is sometimes useful. It's only occasionally useful. And the leaf surface is important. With the center, we can say in Najas telephonian or species without center. in the center is not small portion. It's without center. Indicate the Najas graminium. And here's another character, made flower space. With space indicate Najas orientalis, and then without space indicate Najas graminium. And using these characters, we will say that Najas is the group of 40 species in the world. But sometimes, for instance, without fruits or flower characters, it's really difficult to identify accurately. So that's one of them.
And then I have one question regarding the taxonomy of Najas. The first one is the distribution. And here I listed some well-established species. Then Phylogeny, Graminea, Florida, Indica, Marina, and then the distribution pattern, as you can see, is a bit different. And for instance, some species have really wide distribution, let's say cosmopolitan distribution. And then some others have narrowly distributed species like tropical region species, Indica, and I think for the latter is the same or similar. And the African distribution is there. And this one is a bit different. Let's say a tropical old world distribution. And here is the Najas, is distributed in the Sahara distribution. And for those which have a wide distribution, you could say they might have some cryptic taxa in this distribution pattern. So that's one of the question. Why the narrow species distribution is correct or not. And another question is about the origin of the genus. And this much, I think that the species richness by region, like Asia, Africa, Central, South America, Australia, Europa, North America, just divided into 6 regions. And we have 2 resource divisional work. The first one is the render in 1899, and the other one is trees 1988. And this one is for a worldwide number, and this one focuses on the old world, so no American species. But using both, these are of the dataset, and it says we say that Asia is the most species richness, rich area, and then probably indicating the origin of this group is Asia. So I have my hypothesis Asia as important area. And the other question is about the polyploid. This group is polyploid group, and including diploid, tetraploid, and higher haploid level. And I reviewed some literature from number of Najas, and then listed here. And the red color means the species which have infraspecific polyploid. For instance, the Phylogeny, the literature says that it's diploid, tetraploid, and hexaploid. And similarly, Graminea and the Marina, or oranges has some ploidy differences within the species. This is, by the way, one of the chromosome image of the Najas genus. And if we believe this literature review, we imagine that there is a lot of many polyploidization events in this group. But this is to be revised. Polyploid evolved repeatedly or not? Another question. And to answer the 3 questions, I would have the phylogenetic framework. So let's see how the phylogenetic study of Najas. And among that, this is the most famous and important one, which is in 2010 paper by Donald West and then his colleagues. And this one is the phylogenetic tree of the genus Najas. And when we compare the taxon sampling of West et al. 2010 and the monographic work, their taxon sampling cover only a part of the species, for instance, the subgenus Najas, and then as well as the 4 sections of the other subgenus, 2 of the 4 sections. And at least this species and nudae are not included in the taxon sampling. So we aimed using densely stamped phylogeny to assess taxonomy and the biography and the quality of the genus Najas. So I briefly explain the materials and methods. And then this map is from the, our website, GDIF website, showing the collection, specimen collection record of the Najas. So the color doesn't indicate the species. This just indicates the number of specimen collections. Anyway, and then I had some intensive field work in Japan and Thailand, Myanmar, India. And then for those which are not, for other specimens we collected from the herbarium or previous studies. And then the total number of species is like 9 Asian species, 4 Australian species, and 2 African, 4 European, 4 North American, and then 2 Central and South American species. So our taxon sampling comprises 50 specimens from 80 species. And I just added this video slide to let you know that how we work for the aquatic plants study, because you are not familiar with aquatic plants. And this is some scene of the field work in Myanmar. And we somehow find the, how can I say, this kind of bamboo boat in the lake. So we use this, but sometimes this is the occasion. And mostly I use this kind of stuff and then break the aquatic plants. Okay, and the method of choice. The first one is the chromosome observation. This is kind of basic, orthodox one, no need to explain. And the DNA markers will be cluster DNA, mark KR, VCR, DUV, and DUC1 combined and used. And then nuclear ribosomal DNA ITS. And the phylogenetic reconstructions are using likelihood method and data method. And then finally, ancestral state or area inferences are by the software mosquito or software last. Okay. Now let's see how the genus Najas taxon devised. And then what you can see here is the molecular phylogenetic tree based on cluster DNA and nucleides. And then maybe you can see the similarity between the trees, and then no significant topological difference in congruences that we found. And for instance, that over there, oh sorry, by the way, this black dot indicate the higher slab support, indicating that grade is well supported by statistically well supported. And then over there, you can get the similar topology, and then yeah, the similar topology. And over there, include the species of Najas marina. Now, as you can see, the Najas marina is divided into 2 lineages. So I would say Najas marina alpha, Najas marina beta. This means we need taxonomy to is focused on this. And here another one, the species Najas quadrillata, is clustered with American group. And then here we got the high support for here, and including some species named Najas indica, Najas Inuis, and Najas Ino. We don't really clear about this species because it's here is clustered together, but it's not clustered in the cluster. And the other species include Najas Brassima, which include a specimen from Europe and Asia and North America. And then this includes some different names like Najas Brassilima, Najas Tenuifolia, and Tenuissima, and Japonica, Jasensis, many names. So we need to do some taxonomy work as well. And the king is here, just not sure. And then the problem, the difficult group is what I call the Najas graminea, including many lineages, and it's not really completely congruent to each other. And the significant inconsistent found between the trees are like three specimens. Najas, this one, these two specimens, and then this one. Here, for instance, this one, Najas tenuifolia is clustered with this group in clustered DNA, but in here, ITS3 is placed here together with another group. And similarly, these two specimens placed a little bit differently between the data set. This might indicate the hybridization or early subsequent polyploidy. So here, the insight into Najas taxonomy phylogeny, cryptic taxa including such as Najas marina, Najas graminea, and the hybridization or some kind of polyploidy. And then here, what I show you is proportional tree of the cluster DNA analysis, and then makes ancestral state reconstruction. And this one shows the original result, and then you can see the payment here. And then color indicates the region, and for instance, red indicates Asia, and then blue indicates North America, and light blue is South America. And then some Australian and African species specimens actually included in this kind of grade. And as you can see, the ancestral area of this group is most probably Asia, and yes, it's most probably in Asia. And in addition, we found some important insight into the distribution pattern, species distribution pattern. For instance, the first one, Najas marina, used to be a same species, but now we can say the species include two cryptic taxa named Najas marina alpha, Najas marina beta, and then look at the species distribution pattern. Over here is alpha, the other one, this one is the beta, and we couldn't find any clear geographic distribution pattern. So it needs some kind of species level differentiation, maybe. And on the other hand, we found the interesting distribution. The first one is Najas foverata, which used to be tropical Asian species, but now our analysis includes the specimen from Europe, I think Italy, indicating that we need to revise the species distribution of foverata, including in the taxonomic division of the species in Europe. And here is another thing, Najas nitida, which used to be a tropical Asian species, but now include the African specimen, which was originally identified as the African species, Najas horrida. So maybe we need to compare Najas horrida and indica carefully. So the insights into the biogeography is the Asia being important, and the species distribution, probably taxonomy needs to be revised occasionally. And this is the last one, the polyploidy. Here is the original design here, and then I modified the bigger one. And then the diploid is colored red, and the tetraploid is blue, and the octaploid is it's not clear to this, so American blue. And then we did the ancestral state construction, indicating that the diploid is the ancestral state condition. And then tetraploidization occurred here. And then here, the American group, the branch of the American group, the multicolonization might occur. And then here, Asian group such as Najas indica, Najas canadensis, and Najas kingi. These species could be clade or tetraploid clade, and then this Najas kingi and Gracilima may produce another diploid, tetraploid clade. And the thing is that unfortunately we don't have any chromosome count for the species including placed in this clade, Najas gramínea, but the literature review says that hexaploid is recorded from the species of Najas gramínea. Probably hexaploidization might occur somewhere at the base of this clade. So I would say that we found multiple polyploidization, but compared with the previous literature review, the polyploidization is not really complicated. A summary of today's study. We studied the genus Najas aquatic group in terms of taxonomy and biogeography and then polyploidy. The insights into the taxonomy is the cryptic taxa included in species like formerly widely distributed species. And then hybridization or probably polyploidy may occur. And then in terms of biogeography, Asia seems to be an important area. And the species distribution is occasionally need to be devised. And the taxonomy needs to be defined. And finally, for polyploidy, you say that multiple yet not complicated polyploidy characterizes the evolution of genus Najas. That's a simple evolution. And acknowledgement due to the founding Chinese Academy of Science is one of the Botanical Gardens. Colleagues in Argentina, Thailand, and Japan. And then the following overview let us use the space in the collection and the overview. Thank you for listening. That's all.
Thank you.